Good morning, myself Divyendra, registered research scholar from University of Kolkata, and I have more than 5 years of teaching experience as a Neet faculty. So today I am going to discuss on the molecular basis of inheritance, which includes the structure of DNA, nucleosomal model, semi-conservative nature of DNA, replications, transcriptions, translations, and DNA fingerprinting. So this is the basic chapter in molecular biology and one of the most important topic for the NEET exams. As you know, according to the previous year's questions, so this chapter carries 38 to 42 marks in the examination. So let's start with DNA. So now the question is, what is DNA and how DNA formations occurs in the cell? So DNA is the acidic in nature and it is the main hereditary element in our body which is present in our chromosome. So DNA is a deoxyribonucleic acid which contains 4 types of nitrogenous bases. Number 1 is adenine, guanine, cytosine and thymine. So just look here. So nitrogenous bases. So there are 4 types of nitrogenous bases: adenine, guanine, cytosine and thymine. So in the short, adenine is represented as A, guanine is represented as G, cytosine is represented as C and thymine is represented as T. So keep it in your mind, always adenine can base pair with the thymine and guanine can base pair with the cytosines. So adenine can form a double hydrogen bond with the thymine and guanine can form the triple hydrogen bond with the cytosines. So now the structure, what are the structure? So structure is this. So this is a double helical structure. So 2 strands present in this structure. So one runs from 5 prime to 3 prime and another runs from the 3 prime to the 5 prime. So in this structure, these nitrogenous bases are present. So in the one strand, suppose here A present, so in the parallel strand or the opposite strand, here must be the T. So this adenine and thymine form the double hydrogen bond between them. And after that G, triple hydrogen bond C. So adenine can base pair with thymine by double hydrogen bond and guanine can base pair with cytosine by triple hydrogen bond. So this structure carries, and this is the double helical structures of DNA which was first discovered by the Watson and Crick, scientist Watson and Crick. So into this double helical structure of DNA there are some proteins. So I said earlier that this DNA is acidic in nature, so there are some basic proteins which hold this double helical thread or the structure, right? So what are the proteins? So they are the histone proteins. So histone protein, what are the histone proteins? Histone proteins are the basic proteins. So there are 4 types of histone proteins. So one is the H2A, H2B, H3 and H4. All are in dimer condition. All are present in the DNA as a dimer condition. So there are 4 types of present, and I said all are in the dimer condition. So the total number of protein is 8. So the histone proteins are always octametric, right? So how these proteins present in this structure? So this octametric structure and DNA thread wraps on it. So the DNA thread wraps on it. So basically this is a structure of DNA and histone proteins present in this structure like this. So look at this structure. So this structure is called the beads on string. Why beads on string? Just look here. So histone protein just like a beads and DNA thread just like a string, and this histone protein present in between the DNA structure like this. And you know the DNA base pair which is present on the histone protein is 2000 base pair, right? So base pair, what is the meaning of base pair? So base pair means this. So one nitrogen base can pair with another nitrogen base, and in these conditions adenine can only base pair with the thymine and guanine can only describe with cytosines. If there is any mismatch, then mutations occur. So this is in the later case I will describe. So now this histone protein along with the DNA forms the nucleosomal structure. So nucleosomal structure again compact and supercoiling to form a chromosome, a metaphase chromosome. So now look here. So this DNA can form another DNA, the same copy of DNA. So this is called the replication. So when a DNA makes its own copy from the same DNA, then it is called the replication. And from this DNA, RNA formations occur. So there are three types of RNA. One is rRNA, another is mRNA, and another is tRNA. So ribosomal RNA, mRNA is messenger RNA, and tRNA is transversal RNA. So which type of RNA formations occurs from here? So there are only the messenger RNA. So mRNA formations occurs from the DNA, and from mRNA protein formations occurs. So you know, so DNA to mRNA. So this step is called as the transcription. So this step is called as the transcription, and from mRNA to protein formation is called as the translation. And this whole process, DNA to DNA, DNA to mRNA, and DNA to protein, is altogether called as the central dogma. So this is called the central dogma. So this is the brief descriptions of, or the short summary of the replications, transcriptions, and translation. So there are so many things which I will describe in my later sections. So there are some types of enzymes, how DNA replications occurs into the cells, how transcriptions occurs into the cells, both in prokaryotes and in the eukaryotes. What are the enzymes responsible for this process, and how protein formations occurs from the mRNA. What is codon? What is genetic code? How amino acid formations occurs? So these are the long story. Now look here. This DNA acts as a transformative element. And this was proved by three scientists. First is the Griffith. So scientist Griffith showed that there are some elements which can transform from one generation to another generation. And scientist Griffith showed by an experiment in a mice model, and showed that the one element which is present in our body which can transform into another organism, right? Next, scientist Avery, MacLeod, and McCarthy, they proved by the biochemical experiment that the only, so among these three things, DNA, mRNA, and protein, only DNA can transform from one organism to another organism through a biochemical experiment. And later, Hershey and Chase proved that and showed that only the DNA can transform from one organism to another organism by bacteriophage model. So through these three experiments, first is the Griffith, next Avery, MacLeod, McCarthy, and third is the Hershey and Chase. They proved that DNA is the only element which can transform from one organism to another organism. Next, this replicates through the semi-conservative model. So this semi-conservative nature of DNA was first proved by the Michelson and Starr. The details of this experiment, Michelson and Starr experiment, Griffith experiment, McCarthy experiment, Harsh's experiment, will be discussed in the later sections. So this is the short summary of this chapter, that is the molecular basis of inheritance. So this is a long chapter, so cutting to the short, I will here describe the short summary of this chapter, and there are so many things which you have to read from this chapter, and according to the NEET exam pattern, there are some type of certain questions just like this. So in the question they give suppose they provide this sequence 5' to 3' this sequence. Now they question what will be the mRNA sequence. So suppose this is a DNA sequence. So I say that in this double helical structure of DNA, one strands run from the 5' to 3' and another strands run from the 3' to 5'. So in the question they will provide one strand, that is the 5' to 3'. Now they question what will be the 3' to 5' sequence of that DNA and what will be the mRNA sequence. What will be the mRNA sequence after transcription? So I say in this session that mRNA formations occurs from the DNA via transcription process. So what will be the mRNA sequence if the sequence of 5' to 3' of the DNA is APG, CP, GC? So this type of questions they provide in the examination. So thank you. So this is a short summary of this chapter, and brief discussions of this chapter will be carried on in the later session. Thank you.